I'm happy to introduce our speakers for this morning. Our first speaker is Tracy Bale. Tracy is a professor of pharmacology and the director of the Center for Epigenetic Research in Child Health and Brain Development in the School of Medicine in the University of Maryland. She completed her PhD at the University of Washington in the Department of Pharmacology and her postdoctoral work at the Salk Institute. She has received numerous awards for her research, which focuses on understanding the role of stress dysregulation in neurodevelopmental and neuropsychiatric diseases, and sex differences that underlie disease vulnerability in humans, and using the mouse as a model. She is currently the president of the International Brain Research Organization. Please join me in welcoming Dr. Bale.
Thank you, and thank you for that lovely introduction, and good morning, everybody. I want to start off with a huge thank you to Yoel Sadowski for the invitation. I've wanted to attend this conference, and unfortunately, it usually conflicts with the Society for Neuroscience. This year, due to COVID, they canceled their in-person, so I'm so happy to be here and to engage in person with all of you and those online. And today's topic, as Yoel introduced, is mental health. And so as it is important, of course, to women's health overall, my first comment that I want to start off with is that mental health is health, and that we need to stop segregating it into some special category, whether it's policy, insurance coverage, or access, that mental health is health. And it's so important for women's health, all of our health, especially in the time of pregnancy, as mental health so seriously affects mom and baby's development. So as Yoel mentioned, my lab is interested in stress or trauma because that is so critically important to mental health. So today, I want to engage you in a slightly different twist of discussing the potential understanding of the biology underlying our risk and our resilience, and some of the biomarkers that we'd like to explore. I'm going to tell you two small parts of stories from the research going on in the lab, but really only in part to convey the necessity for understanding the risk for mental health disorders. So it is clear that during the pandemic, there we go. That stress we are all experiencing, again, being here in person does not come without certain risks, and that the CDC is tracking our levels of reported, or increased levels of reported stress and anxiety and depression. As you can see here, these are actually data only as of December 2020. I looked for them quickly this morning. They haven't updated the graphs, but you can see the fast rise in reported cases of anxiety and depression that are escalating during COVID. And we know that in our most vulnerable populations, these numbers are skyrocketing, as you can see here from looking at the gap by gender. And we know that the increased prevalence of anxiety and depression, especially amongst our women, especially as relationship to those who have young children at home. That even in the academic sciences, we know that the burden of having young children at home is falling not only, but largely, and putting extra stress on our young moms. We're seeing this data in the papers being submitted, the numbers of grants being submitted, and the consequences, I think, that we need to think about of how this pandemic is going to affect many of our colleagues for years and years to come. So as I said, my lab is very interested in studying stress, and stress, as we use mouse as the model. We also do a lot of translational research in human populations. So in mice, we refer to it as stress. Obviously in humans, that could be trauma, adversity, many of the things we've already heard about in this conference. So in studying risk and resilience, I love this schematic, this tree, thinking about the African proverb that the wind does not break the tree that bends, meaning the resiliencies, whether they be genetic resiliencies, whether they be social support. In fact, if you look at the root system of this tree, I like to call that the social support network that we all have or don't have. And then again, in many of our most vulnerable populations, it is that social support network underground that is missing. And how do we understand those contributions in the face of adversity of who is most at risk and who is most resilient? I was also recently at a conference, as was introduced, that I'm the president of the International Brain Research Organization, so I was just in Africa for a conference. And I was also told when I showed this slide of another African proverb I want to remind everybody in this room of, which is that be humble, because even a large tree can be taken down by a very small axe. So who is at risk and how do we define it? I like to give credit to Tom Insel, the former director of the National Institute of Mental Health, for really driving home this idea of the G by E by D. In many diseases, but especially in those that affect the brain, it are very complicated. And we need to think about these in ways that are not just simple, it's stress or it's a dietary concern. It is all of these factors that are so critical when we think about our models and mechanisms. The G by E by D, your genetic background that you inherit from your mom and your dad. We talked a lot about those yesterday, especially as Dr. Hood introduced in yesterday's session, thinking about all of our genes that together contribute to our risk and our resilience. By E, that's the environment, the experiences and exposures that happen, the adversity, the good things, the bad things, and not just which experienced once you're born. That would happen during pregnancy, and I'll show some data today on that which even happens in the preconception environment. The experiences that your mom and your dad and other generations bring to conception and how important they are. And then the D, those developmental windows, when that adversity or resilience was wired in. Did something good or bad happen to mom while she was pregnant? Did something happen at birth? Did something, as I said, happen in the preconception environment? Did something happen during puberty? This is a dynamic period of maturation for the brain. These are all important to understand, and they are complicated, but they need to be thought about because the simple models are not moving us forward in the field of psychiatry. And underneath all of the G by E by D is sex as a biological variable. And this really, I think, was driven home by Janine Clayton and the Office of Women's Health Research when they said years ago, if you're submitting an NIH grant, you need to address sex as a biological variable. And what that did is it opened up a lot of individuals to start thinking more about the differences between men and women and males and females. The sex chromosomes, the sex hormones, and as I highlight underneath here, sex as a biological variable touches upon G by E by D. The chromosomal genes that might be specific to X and Y chromosomes, the environments that we might be experiencing, how we perceive them, and of course, it's going to be different in the developmental windows for males and females all along the lifespan. So how does this all impact more than one generation? And thinking about the adversity piece, the Kaiser Permanente studies from the 90s highlighted for us that adversity in the lifespan, and I like to think about the ACEs, the adverse childhood experiences, those that occur before you would ever conceive a child likely, are so important for determination of disease risk across the lifespan. And they determined from those studies, albeit largely done on a mostly white population, and I'll come back to that 4 or more ACEs prior to the age of 18 was the strongest determinant for many disease risks, many disease risks. So if you look on the other side, the left side of this slide, you can see that what diseases related to neuropsychiatric disease you might think about being very impactful across the lifespan, and especially for pregnant moms, to impact mom's health and baby's health, and altering the course, especially of brain development, that might place that child at a greater risk. So thinking about these ACEs and the adverse experiences, you have to think about specific populations. And as I said, the Kaiser Permanente study was done on mostly white population. And now there are great studies coming out looking at these high-risk populations, our most vulnerable populations. So in understanding pregnancy and how I think in our lab about modeling stress, we have to really understand the population we are trying to model. And so in a study that was recently done on the Department of Psychiatry at the University of Maryland, they assessed across the US currently pregnant women and the number of ACEs they experienced. So before they turned 18, the number of ACEs they'd experienced. And as you can see, they're highlighted in yellow, that 4 or more cut point of risk is about 12.5% for most women on average across the US. I live in the city of Baltimore, a majority black city that has been exposed to enormous amounts of racial discrimination over many generations. Do you want to guess the percent of women in the city of Baltimore that have experienced 4 or more ACEs? It's over 60%. So currently pregnant women, The majority in the city, the majority Black population of Baltimore, over 60% have experienced 4 or more ACEs. And I highlighted on there, this article just came out, that the family of Henrietta Lacks, to highlight the generations of discrimination and medical mistrust. And so when you, as I was introduced, I am the director of a center at the University of Maryland. We engage in the community to understand the adversity and risk, to try and understand how we might identify things like biomarkers, how we might address issues with children and brain development, et cetera. And it is an enormous task because of these generations of medical mistrust that have been built in. So again, understanding our communities and understanding the factors that influence risk, we need to understand the populations we are attempting to model. Okay, so I am a neuroscientist, and I am exceptionally interested in defining mechanisms. And so it's a huge leap that we face, especially every grant that I sit down to write, in that I want to understand the molecular and cellular mechanisms that define risk and resilience, especially for the brain. The brain is not in isolation, so we must understand the whole organism. But in order to really understand the route at which the brain takes in development, we must first understand what is brought together when sperm and egg meet. And so we have many grants and studies in the lab that focus on the preconception environment. And I don't have time today, of course, to go into them all. But what I do want to focus on, that mom and dad have lifetime experiences that they bring together at the point of conception. And these don't have to be dramatic cellular mechanisms. They can just be subtle. They can be subtle changes at the rate at post-conception, the rate at which the development of the embryo occurs, the rate at which implantation occurs. All of those factors can be very subtle, but can compound over time to alter how that brain is developed, missing key periods of time where critical pruning of important synapses may occur, as an example, going back to that G by E by D. So we don't need to have really dramatic effects. We can have very subtle changes in cellular mechanisms that can lead to increased risk or resilience that we need to understand. So this slide is just to remind everybody in the room that in order to understand mom's health, and especially baby's health and brain development that put us at great risk or enhance resilience across our lifespan, there is a very complicated order of things we need to understand. In order for that brain to develop, we need to understand mom and dad's preconception. Those are the germ cells on here. We need to understand how those changes might be influenced by the current intrauterine environment of mom that might be influencing things like the placenta, which we heard a lot about yesterday. And of course, the stage and the course of pregnancy, whether mom is experiencing dietary challenges or infection, or adversity and stress, or previous trauma. All of those factors come together and are critically important in our models to understand how the brain is being developed. Okay, so now I want to segue into this idea of biomarkers, because one of the best ways we can take our bench research, our studies done in mice, which of course we heard some skepticism yesterday, and I'm on board with the fact that a mouse is not a human, but I have also highlighted that some of our best ideas and understandings of mechanisms have been done in model organisms and taken back in, translated back into the human, and then back into the mouse again. And my lab spends a lot of time thinking about that. So extracellular vesicles are nanoparticles, and you in this room, especially from the Magee, know a lot about them. Yoel Sadovsky, I think, is the world leader in really giving us information about the importance of extracellular vesicles. I'll come back to a lot of Yoel's work as it relates to the placenta especially. These nanoparticles, within the last several decades, as you can see in the bottom of the slide there, we've learned more and more about what these are. They are secreted from every tissue in mammals, including the brain. They can act locally and they can act at distant sites. They carry messages of things like signaling to the immune system. We've actually shown lots of data that suggests that they are a component that tells you a lot about stress in the environment. So our understanding of what these are, and a term biomarker, it could be a current state, it can tell you about the history of the individual, which I'll show you data on, or it may tell us something about the signals that the body is trying to compensate for that may also involve engaging the brain. They contain lots of proteins in the lipid membrane. The inter-nanoparticle compartment contains a lot of small non-coding RNAs. So they deliver a signal, they can alter function ultimately, and they travel in high concentration in circulation. In the lifespan, the time at which they are at the highest concentration is pregnancy, and we'll come back to that. There's different kinds of extracellular vesicles. Many of you have probably heard of them called exosomes. Exosomes are a very small size of extracellular vesicle. There's also microvesicles and other types of vesicles. They're different in how they're secreted from the cell, and they're different in how they are compartmentalized and from where they are secreted from. So I'm going to tell you a little bit of a story that just came out in biological psychiatry. This is work that was done in the lab by Katie Morrison, who's actually sitting right over here. She was a postdoc in the lab. She is now an assistant professor at West Virginia University. So we did this in collaboration with the Grady Trauma Project. The Grady Trauma Project is out of Emory University in the city of Atlanta, also a majority black city. And in this study, Kerry Ressler, when he was a resident, a psychiatry resident, started this most amazing program. He decided, in looking at this inner city hospital, why are we not capturing all of the information for all of the individuals that are coming in, especially to our emergency room? Now, Kerry is particularly interested in PTSD, and as a psychiatrist, trying to understand the factors in the lifespan that contribute to PTSD risk. So he started the Grady Trauma Project, and then it was picked up when he left for Harvard by Tanya Jovanovic. Tanya is now at Wayne State starting a Detroit trauma project as well. So what this does is the Grady Trauma Project captures everybody. They enroll everybody who comes into the emergency room. They capture all of the information from their lifespan, the adversity that they've experienced. They get biological material from them, and they enroll them often in studies, including looking at PTSD and other factors related to psychiatry. So we wanted to ask a very specific question. We know that women who've experienced trauma or adversity in particular times prior to the turning 18, right, those ACEs, but really dividing it into more specific timing, because we know the brain develops and matures over the course of childhood, and when adversity occurs, surely must be impactful for what you are at risk for later in life. So we divided our study. We had evidence from our mouse models that this mattered. So we joined forces with Tanya Jovanovic and her group, and we asked, could we get information from the Grady Trauma Project where we would take women who had experienced a particular type of interpersonal violence, sexual trauma. Why sexual trauma? Because sexual trauma we could define by very specific periods of when it was experienced with relative accuracy, unlike asking more about overall trauma. So these women in the city of Atlanta, it's a majority black city. It's like a 98% black cohort that we have recruited for this study. These women are in their late 30s, early 40s, so this is many decades after the traumatic experience has happened. There is an overall large amount of PTSD and trauma equally divided amongst the groups. These women are divided into experiencing sexual trauma under the age of 14, during adolescence, defined as 14 to 17, or as adult, or no sexual trauma at all. And again, all the confounds of PTSD, symptoms, treatment, and overall trauma are normalized across all of the groups. We wanted to ask, if we take biological material, we take a blood sample from all of these women currently, despite the fact that it's been decades since the trauma occurred, could we isolate extracellular vesicles, and doing an omics approach, which we talked about yesterday, could we assess any factors, variables, find any proteins or small non-coding RNAs that could guide us in understanding that there might be something programmed or a way in which some specific tissues were engaged in risk for these women. We also assessed a bunch of other parameters I don't have time to talk about today. Okay, so first and foremost, extracellular vesicles. So these can be very informative just by doing an initial assessment and understanding the size and composition of the extracellular vesicles. They can tell you a lot about the fact that something systemically has changed. So I just want to show you quickly two pieces of data that Katie found in these extracellular vesicles. And again, thinking about them as potential biomarkers. On the left side is looking at the median size. On the right side is the zeta potential. So in assessing the entire population of extracellular vesicles from circulation in these women in their late 30s, early 40s, we're looking at the size. This is done by nanocyte technology using Brownian motion to tell you about the overall size parameters of extracellular vesicles. So what the size tells you, why is this important? It can tell you a lot about the endocytic rate and the EV type, the extracellular vesicle type. Are you shifting many decades after a specific traumatic event happened? Are you shifting the population? And then if you could narrow it down a bit, the particular tissues involved, you might be able to assess a greater understanding of risk for this individual. So as you can see, we've divided this out by no interpersonal violence, under 14, 14 to 17, and then over 17, meaning 18 and up. And as you can see, we see a significant reduction in vesicle size in our adolescent exposed group. What that tells us is that we're driving our vesicle population more toward an exosome-like vesicle, and that may tell us something specific and unique about the population of EVs being secreted that we can then follow up. On the right is looking at the zeta potential. The zeta potential tells you the surface charge, which is built upon both the size of the vesicle and the protein content, and it has a lot to do with the way in which vesicles interact and the slipping plane, which I'm not going to get into the physics of, but it has a lot to do with how two vesicles may interact with each other or another cell type. And a way in which the zeta potential of the charge is really important because it tells you a lot about the stability of the vesicles in circulation and their adsorption. So the more negative the charge, the more stable in repelling vesicles they are and less likely to aggregate. And what we see, again, in this one particular population, the women from 14 to 17, those adolescent exposed women, had a reduced zeta potential, meaning that they have a smaller vesicle size and more likely to be aggregated and degraded. So a more dynamic population of extracellular vesicles to follow up on. So the next thing we wanted to assess was really if we did a proteomics approach, could we assess, we're not changing, there we go. If we wanted to assess the content of these vesicles, we know we're shifting toward a more exosome-like vesicle, is there a difference in what the signal is that is being relayed? Can it tell us something about the tissues the vesicles are more or less coming from? So I don't have time to go into all of the data here, but what I want to highlight in this heat map on the top is looking at no sexual trauma compared to all sexual trauma grouped together. Blue is a reduction heading towards the red or pink, blue is an increase in a certain type of protein. Pink or red is going toward the reduction. And you can see that there is a wide array of detected proteins. You would expect that, because remember, they're secreted from every tissue in the body. If you then parse out the differences below, you can see again the timing of when sexual trauma happened. You can see in this one particular group here, this very high, sorry, this is not changing. There we go. You can see in this one particular group here a large number of proteins that are very highly expressed, again, only in that one group of women. And so we wanted to know what those proteins were, and could they tell us something about the integration of the systemic system of secreting EVs into circulation, either where they might be coming from or where they might be traveling to. And so what it turned out, which surprised us greatly, what these EVs are, is they were a lot of keratin and keratin-associated and related proteins. Initially, we thought, what is this? What are keratins doing in EVs? When we further assessed and looked at the examination across all of our groups, it turned out that these keratin-related proteins were not only highly expressed in this group, for the most part, they were only expressed in this group. And you can see that in that bubble plot in the middle there, all of those proteins abundantly expressed in EVs and only from this one group of women. What was further intriguing as I began digging voraciously through the literature, trying to figure out what is the commonality of these things, and thinking about a grant, of course, it turned out that the majority of these proteins, their genes are part of a specific 17q21 gene cluster in humans. And that is interesting for anybody who studies gene regulation, because that gives us a potential mechanism whereby an epigenetic change that regulating this gene cluster may have been altered during that traumatic experience in adolescence. What is also of great interest, and in the review of this paper, one of our brilliant reviewers highlighted for us that also in this gene cluster are the CRF receptor 1 and MAPTau, which are both genes identified in a GWAS for risk for PTSD. So within this gene cluster, we have a lot of genes now that are showing up in our extracellular vesicles, suggesting a common point potential of traumatic experience. But what do they mean? As a neuroscientist, we often don't think about the skin whatsoever. But it turns out, as I was reminded, their skin is your greatest barrier from the environment. It is highly innervated. It is autonomically innervated, very much integrated into the system by which things like PTSD evolve. And it also turns out that the hypothalamus pituitary adrenal axis, your stress response axis guided by your brain, very similar system exists in the skin. The same molecules, the same receptors, and the same response. So then it left us with the question of, well, what is specifically happening in terms of autonomic regulation in the skin that could be guided by some experience in adolescence? I dug a little further. And relevant very much to a recent Nobel Prize that was just given out. It turns out that a lot of our genes that were found in that cluster are actually relevant for what's called a Merkel cell. So the Merkel cell, what is a Merkel cell? And so these are touch domes within all mammals that are important for light touch. And you'll recognize this again from the Nobel Prize that was just handed out. So Merkel cells are actually mechanosensory skin cells. They respond to light touch. They're in very targeted places around your skin, and the skin of mice as well. They respond to light touch. The piezo-2 channel was recently just identified as the channel, as the transducer of that touch in these touch domes that then allows the activation and release of norepinephrine. And it turns out Merkel cells are the only neuronal-like cell in the skin, which became fascinating to me as a neuroscientist. So they're only neuronal-like cell. When activated, they release norepinephrine to the A-beta fibers that are innervating these touch domes that feed back to the sensory cortex in your brain. And the final piece that was fascinating to me to try and link this all together of how this makes any sense, the final maturation step of Merkel cells occurs during adolescence. So putting all this together from an unbiased bioinformatic omics approach, we've now begun to identify a potential risk time during adolescence where a specific type of interpersonal violence may place these women at greater risk. A risk for what is a question. So I don't have time again to go on all this data, and it's all published. You can look it up in biological psychiatry. But the timing sensitivity, we also wanted to know, of course, risk for what? And so in identifying this, we looked at things like risk for things related to obesity, diabetes, and hypertension. So on the left, you can see when we parse out our data, just like others have found previously, sexual trauma and interpersonal violence early in childhood or during adolescence puts you at much greater risk, especially women, for obesity, diabetes, and on the bottom you see a greater risk for earlier entry into hypertension risk. But on the right, interpersonal violence of sexual trauma during adolescence, that one specific group, puts you at much greater risk for presentation of PTSD as measured by fear-potentiated startle and skin conductance. So there is, again, back to that skin thing. And I just want to highlight by reminding everybody the terms that we use. He got under my skin. He made my skin crawl. There is a reason why our skin is our greatest barrier, and understanding its innervation is very important. So I just want to bring this all back to pregnancy. If you are now a woman who's experienced those particular types of trauma at particular time periods, and you are now pregnant, trying to understand the risk factors and maybe identify the biomarkers of risk, coming back to Yoel's work, thinking about the placenta as it secretes an abundance of extracellular vesicles into circulation. If we could understand the pregnancy period and maybe identify these similar biomarkers, understand the types of vesicles secreted, integration with the systemic, autonomic, nervous system, brain, immune system, we might be able to understand risk. And I'm thinking risk especially for our most vulnerable populations again. As we heard yesterday, thinking about maternal death rates, especially for Black women, which remain very high in this country, as well as mortality rates for babies in the first year postpartum. And again, I want to highlight some work that was done by another postdoc who was in the lab, who is now an assistant professor here at the Magee, Eldon Juserovich, and understanding how do we model these complexities in mice and thinking about this. And Eldon has put together a model. We're using dietary challenge and maternal infection and stress and able to model that mortality rate. And as you can see here from a mouse model that he developed, increasing the mortality rate within 48 hours of delivery to 60%. So again, how do we understand these mechanisms, and then how do we translate them? And so I just want to end quickly with moving from mom to dad, because as we heard yesterday when Yoel started this whole conference and thinking about it, the health of everybody, including dad, is important to the health of mom and baby. And so how do we understand the sperm? And I want to use just a quick excerpt from a study that came out last year in collaboration with Neil Epperson, who's the chair of psychiatry at Colorado. We wanted to assess dad, because as I'm talking about these biomarkers, we really needed to understand mechanisms. Again, going back into the human so we can go back into the mouse. And we wanted to understand, as I'm talking about these small non-coding RNA coming from extracellular vesicles, we had been doing a lot of work on paternal stress in our lab with mouse models. I'll show you in a second. But no one had done a full assessment of the composition of what is in sperm and delivered to the egg. And without doing a whole assessment, without doing a whole bioinformatic workup, how are we ever going to be able to assess risk? Knowing what dad is experienced and what is delivered outside of dad's DNA. So I'm not going to go into all the details. Again, this is all published work, but I want to put out this provocative idea of dad's lifetime experiences as well, carrying signals to the conception, to the egg, in addition to his DNA, can shift the course of conception, implantation, and development. And we know this from these studies that we did on a relatively healthy population at the University of Pennsylvania when Neil and I were both faculty there. And we recruited in these undergraduate and medical students. We paid them handsomely to come in once a month for 6 months so we could assess the content of their sperm over the course of 6 months, because we did not have any idea what normal sperm looked like in terms of small non-coding RNA content, to then be able to ask. So Neil's psychiatry group did a full workup on these individuals and did a stress assessment, ACEs assessment, psychiatric workup every month on the individuals and asked about perceived stress. We're looking for a dynamic state in these sperm. And so this is the very, very complicated data, which obviously I'm not going to go into. We assessed microRNA, we assessed tRNA, and we assessed piRNAs. And every column is a different individual. This is just showing you 5 of our sample, showing you the very dynamic population of these small non-coding RNA. And when we did a complex mechanism where we worked with the Institute for Genome Sciences at Maryland and asked, if we take all of these microRNAs, tRNA and piRNA, and perceived stress and ACEs experience, and we took about 1.5 years to do this and model all of this, what can we learn? It turns out we can learn a lot. And so within each of these categories, we identified in a time plus 2 months, if you experience stress that reaches a certain level, we will see a change in certain small non-coding RNA in your sperm 2 months later. And interestingly, of the microRNA we identified, 2 of them are well known to be important for fertility and pregnancy. So stay tuned for where those data will take us. And I just want to say that these studies have then taken us into understanding the mechanisms by which understanding the somatic programming, taking the data from the human back into the mouse so we can understand, where does this happen? How does this association change with stress experience end up in sperm? Right? It's not magic. It doesn't just happen in the sperm, obviously, because the sperm are all packaged. This is not happening. It's when the sperm are traveling, leaving the testis and traveling through the epididymis. We now know in this field that the extracellular vesicles, as I told you, they're secreted everywhere. Those extracellular vesicles secreted from the cells surrounding in the epididymis, these epididymal epithelial cells, interact with the sperm and are carried to the egg. And that is the way that dad's dynamic environment can shape and shift. And the reason we know this, because a very brilliant graduate student, Jennifer Chan, modeled this, and her thesis took years and years of work to figure out how to collect vesicles from epididymal epithelial cells. She called this stress in a dish. And this is all published work. You can also look this up. And she was able, in a brilliant experiment, to ask about causality, because that is our obligation as bench researchers, to ask the causal questions. She was able to model the stress in the dish, collect the extracellular vesicles in 2 populations, labeled here as vehicle versus corticosterone, which is the stress hormone. She was able to take sperm from 1 male, so we're controlling for dad's environment, divide it into 2 pools, incubate it with those vesicles. She was able to take eggs, perform ICSI, or intracytoplasmic sperm injection, inject that sperm into 2 populations there, take the same donor mom, put them into the left side or right side of the uterus, grow them up, ask about mid-gestation development, or look at the animals as adults, and boom. She was able to causally show that it matters. That stress signal in those extracellular vesicles changed brain development. By mid-gestation, their brain is developing faster. And by the time that these animals are adults, their stress response is now different. So I'm going to leave you with that because we're out of time. I want to thank, of course, the lab members who contributed to this work. I gave them credit throughout the talk. I want to especially thank NIH for funding all of these studies and all of our collaborators that I mentioned throughout. And thank you for your attention.